Good morning, everyone, and welcome to today's session. My name is Marissa Fraser, and I am the Marketing Communications Manager for Pion Inc. Today's presentation title is Advancing Vitro Methods in Generic Drug Product Development, and will be presented by Joseph Baranek and Teresa Kretschy. Joseph has worked for Zentiva since 2011, and he currently leads the pre-formulation and biopharmacy units within Zentiva R&D. He is involved in a variety of drug development projects, contributes to the selection and characterization of alternative solid forms of active substance, and is engaged in the selection and optimization of prototypes for clinical studies. He is also responsible for the development of innovative dissolution approaches, pharmacokinetic modeling, and collaboration with external partners and universities. In 2005, Joseph received a master's degree in biochemistry and biotechnology from the University of Chemical Technology in Prague, Czech Republic. And in 2010, he finished his PhD in chemistry from the University of North Dakota for his work in analytical and environmental chemistry. He then worked as a postdoctoral researcher at Pacific Northwest National Laboratories in Richland, Washington, in the group of chemical physics and analysis. Now, Teresa works as a pre-formulation and biopharmacy specialist at Zentiva in the Czech Republic. And in her role, she is responsible for physicochemical characterization of APIs, early-stage pre-formulation experiments, solubility and dissolution experiments, and their correlation with clinical trials. An important part of her job is risk mitigation before bioequivalence studies and prediction of their outcome. Teresa holds a master's degree in analytical chemistry, and she studied her PhD at UCT Prague in the program Drugs and Biomaterials. As a member of the PARC, or Pharmaceutical Applied Research Center, she also worked closely with the industrial experts from Zentiva throughout her studies. In her research, she focuses on in vitro characterization of bioenabling formulations, as well as on their rational design. Welcome, Joseph and Teresa, for being here with us today. We are also going to have our Carl Box, Chief Technology Officer for Pion in Europe, and he's going to be assisting us with the Q&A. So the Q&A, we will conduct a short Q&A session at the end of the presentation. And if you have any questions during the webinar, just type them in the Q&A widget, and we'll address them at the end of the presentation. So we're going to try and cover all questions at the end. And if we run out of time and don't get to answer your question, we will answer it to you directly by email. I also want to mention that this presentation is being recorded and will be available on our website as well. Also, everyone that registered for today's session will receive an email with a link to the recording. So now, without further ado, I will hand things over to Joseph. Thank you.
Thanks a lot for the kind introduction. And today we'd like to take you on a journey through the in vitro methods that we use in our development, which is a generic drug product development. And we really hope that you will enjoy it, hopefully find it useful in some ways as well.
We will actually start introducing what we do in Zentiva development. As you may know, we're a generic company. That means we're developing products with already known and described active substances. And one of the basic requirements for us is to pass a so-called bioequivalence trial in the end of our development to prove that our drug, based on the pharmacokinetic profile, will work as well as the original or reference drug product. So here it is depicted on this picture here, and here is the result that we usually aim to obtain, that is a bioequivalent product on Cmax and AUC. And what do we develop? One of the pillars that we focus on is the products that are soon to have the patent protection expired, and that means that the generic players can enter onto the market. And our aim typically is to enter the market as soon as we can. And what's interesting here for this generic development is that we typically need to overcome many competitor patents. Typically, patents are preventing us from launching a drug product which would contain the same polymorphic form of API, or maybe limit us in the composition that we use, or possibly technology that we can use to produce tablets, and that's simply something we need to overcome. And our second pillar here is what we call life cycle management, or cost reduction products. This is quite typical for generics. We're trying to decrease the cost of manufacturing and thus potentially decrease the cost for the payers and patients. And basically, we develop these products typically because of the products which we were developing for the loss of exclusivity market were more expensive, simply because we couldn't use the cheapest possible technology or available form of API, and here we make them cheaper. And the third pillar, also very important one, is our generics with added value. Like on this example, we have a product with the active substance dasatinib that we developed to improve its efficacy, let's say, for patients who have higher stomach pH. Either they are taking proton pump inhibitors, or they have naturally higher pH in their stomach. And for these patients, the original drug product didn't work so well. We actually succeeded to remove this pH dependency, and now our product works, or has a potential to work much better in those patients with higher pH in their stomach.
And now let's have a look what we do in the biopharmaceutical area in Zentiva, what we're focusing on, and what we are actually using some Pion instruments that we have available in our laboratory. So the first thing that we typically look at is the physicochemical properties of the compound or the molecule that we're going to work on, like solubility, dissociation constant, log P, dissolution rate, intrinsic dissolution rate, some of those useful measurements that tell us more about the molecule. Then we also often study the effect of excipients on the dissolution and precipitation behavior. We're interested in how some excipients can stabilize a supersaturated state of the specific molecule, and so on. And we're very much interested what can happen with the drug product in the human body. We look at how the tablet or capsule disintegrates, how it dissolves, and how it can potentially be absorbed. And this should end up in the prediction of the bioequivalence study outcome. We're running some pharmacokinetic simulations. And how do we actually run all these experiments and measurements? So typically we utilize something that most of you probably know. It's USB-1 or basket apparatus and USB-2, or sometimes called pedal apparatus. Obviously, we also have USB 3 or BIODIS equipment, and as well as USB 4, or sometimes called flow-through cell. With all of these, we need to utilize either UV-Vis determination of the dissolved compound, or sometimes we need to use HPLC for the measurement. The advantage of these standard apparatuses is that they are relatively easy to operate, and the measurements are quite reproducible. Uh, there are some disadvantages though, that we typically need to have relatively large volumes in those experiments, need large sample quantity, and often it's questionable how much these measurements represent the behavior of the compound in the human gastrointestinal tract. And that's when the Pion instruments come into the game. The first instrument that we're going to talk about is Sirius T3. This is the one we had actually the longest in our laboratory, and it was made by Sirius when Sirius was still independent company and Pion was an independent company, so we call it Sirius T3 here. It may have a different name now, but the way this thing works is basically an automated titrator, uh, thanks to which we can actually run many samples, maybe overnight or at once. Um, as we mentioned, we typically try to understand the basic behavior of the molecule that we start to work on. So we use this instrument to measure pKa and log-P if the compound is dissociating. Sometimes we utilize it for solubility measurement based on check-sol principles. The big advantage of this instrument is that it works really well even for very poorly soluble compounds, thanks to the possibility to measure the assays either by UV, or when there is something more soluble, we can use the pH electrode for standard pH titration. Besides those two options, how to measure the titration, uh, the experiments can be also run with co-solvents to help solubilize something which is really poorly soluble. And in the end, the the software of the instrument can calculate or extrapolate the values obtained with co-solvents to zero level of co-solvents, and then you obtain dissociation constant, which which you would obtain in aqueous system. On the next two slides, we're going to show a relatively simple example and demonstrate why it is useful to run a pKa measurement even on the relatively known compounds which are already generics, and you would expect that there are information in the literature even on the basic properties like pKa. So in this case, you see the molecule here, and in chemicalize search, we found that the molecule should be able to dissociate on this NH group, and has a predicted pKa value of 8.23. If we search other resources, we will find that the pKa should be 7.2. So now you know how to decide which value is true, and the only way to find out is to run the measurement.
And we can see the result of the measurement here on this slide. On the upper figure, we see the record from the UV detector, or from the UV probe. You see that the two species, so protonated one and deprotonated one, are nicely separated, so we can then easily calculate the pKa value. And what our result is 7.11, which is close to what we saw in other sources, 7.2, but differs from what we found in as a prediction from chemical eyes. And one important information we obtain here, that 7.2 is relatively close to pH of the medium which is used for the QC test, and it's a warning, or it's the saying us, okay, be careful. If you see something strange in the dissolution, it may be because of your medium is relatively close to pK of the compound, and then in this case some weird things can be happening in dissolution.
Here on this slide, we want to show how important it is to measure pK values even for the molecules which are already known and described. What we're showing in this figure, or what we plotted here, are the data measured in our laboratory using series T3 measurement and data which we found in the literature. So in this first example here, you see that there is a relatively big difference, almost 2 units for this particular compound. But then we have some which correspond quite well with the measured, when we compare measured and literature value. But there are some which do not correspond at all, and there are a number of molecules which there is a small deviation between literature found value and measured value. And as you can imagine, the precise knowledge of the dissociation constant can be quite crucial for many things. It is crucial for the behavior in the gastrointestinal tract. If it's a basic compound, it will impact how the compound can precipitate, at which pH it will start precipitating. It will impact absorption in the human body. If the compound is charged, it will probably diffuse slower through the intestinal barrier, and so on. There are many things that pKa value will impact. On this next slide, we want to demonstrate probably not so typical use of T3 instrument and measurement. In this case, we used it to run a small dissolution study on different forms of the same API. And what you see in the figure is the plot of the compound dissolved over time, and we plot it here as absorbance, so which should correspond to the concentration of the dissolved compound. If we look at the result, the first result is for the free acid, form A, which is the most thermodynamically stable form of the API. What you see here, it dissolves to a relatively small amount and stays there, does not move anymore. And then we have a number of different salts here, which they supersaturate to a different level. And also what we observe, their induction time, the time before they start to precipitate, differs. So that tells us and describes in great detail the salts of this molecule, and this result will help us to select the most proper form for what we want to do with it. The advantage of such measurement by series T3 is that we only need very little amount of API to run this. One disadvantage may be that the assay does not allow to thermostat at 37 degrees Celsius, so we run it at room temperature, but still it is very useful and gives us some initial idea about the compound. And now on the next slide, Teresa will continue with the description of the next instrument, which is Sirius Inform.
So the next instrument that we have available from Pan Company in our lab is called an Inform. In many ways, Inform is similar to T3. It's also an automatic titrator. We typically move to Inform once we have a little bit more sample available. So typically we would use tens of milligrams of API for measurements that are done on Inform. Once again, the experiments here can be run overnight. The instrument is fully automated, so we can set up even different assays and well, let them run when we are not there. And if everything runs smoothly, we just come back in a few hours and get the results. The nice thing about Inform is that it has really very well-controlled conditions of the experiment, and it collects a lot of data throughout the experiment, or a lot of online data. One of the outputs is, of course, the measurement of UV spectra by UV dip probe. Here I just added a plot that shows the measurement of, well, this is from dissolution experiment. And you can see how even at low absorbance, we are able to, well, see the whole UV spectra, and eventually compare it against standard measured under the same conditions, which is the black line here, and see whether what we are measuring in the dissolution actually matches our standard. As I mentioned, you can see that it seems to be really precise at even really low absorbances. Also, as for the controlled conditions, because Inform is an automatic titrator, the pH can be easily adjusted throughout the experiment by titration, and it is also collected throughout the measurement. Also, we can, of course, control the temperature and collect the log of the temperature as it was maybe fluctuating throughout the experiment, and the temperature is controlled by a heater block that is inbuilt in Inform. Well, Inform has, of course, many different features, but these are just some of the highlights I wanted to mention here. And now let's move to some of our data. So typically what we would use this instrument for regularly is measurement of intrinsic dissolution rate. For us, you know, the reason why we want to do it typically is to select the best possible candidate in terms of solid form. Well, our motivation can be twofold. First, we might be looking for an alternative solid form to that was used by the originator or by the original company. Or on the other hand, we might be looking for a better solution if you're looking at the projects where we're targeting improved bioavailability. And here at this graph, let me show a typical result of our IDR experiments, or intrinsic dissolution rate measurements, where we screened a number of salts of different API. What we see here is that we got different intrinsic dissolution rate in, you know, but see here you see just a number, and of course we had to evaluate the data somehow. So I'd like to show you also how, well, typical output of the instrument looks like. So let's see here. For instance, this would be our reference form B. That's a form used by the originator, and whenever we're measuring intrinsic dissolution rate, we're looking for a part of curve that follows, that follows basically a linear line here, and where we can apply linear regression and from that calculate the intrinsic dissolution rate. So you can see that for this form, we got quite a nice result. We were able to evaluate the value that we're looking for. Everybody's happy, and now we're looking for either form that is, you know, at the same level, or maybe faster, in case we are looking for something better than the originator has. And when you look, for instance, for the HCl salt of this compound, you can see here that I put a note that this disintegrated, and I wanted to show you how that looks on the data. So when we plot the actual data from the solution, we see that first we do measure something that can be evaluated as intrinsic dissolution rate, and that's actually the number that we plotted here. But then you see like an abrupt change in the data, which corresponds to the disk being disintegrated, and basically the API wasn't dissolving from controlled surface anymore. It could dissolve freely. And one last example that I wanted to show is a typical result that we got for fumarate, for instance, in this case. And you can see again that the calculated intrinsic dissolution rate was somehow higher than form B, which is something that we're looking for, but we have a note here that there was a decreased rate of conversion of this form. So this is what it might look like in the data. We get a fast dissolution in the beginning, which corresponds to the intrinsic dissolution rate of the fumarate. But then as the salt converts to a more stable form, which is in this case actually form A, you see that if we wanted to evaluate the intrinsic dissolution rate in this region, it would be much slower. And the interesting part is that this part of the curve actually corresponds really nicely to intrinsic dissolution rate of form A, which you see here. And that's also the same thing that we measured on XRPD, or on where we evaluated that our fumarate actually converted to form A. The next type of experiment that we usually run on Inform are precipitation experiments. Here we are typically looking for an excipient that would act as an optimal parachute for amorphously dispersions that we might want to prepare. So once again, oh yeah, and this is a picture that you are probably familiar with that depicts this concept of spring and parachute. So whenever we have a quickly dissolving solid, which can be, for instance, amorphous drug form, well, it has a tendency to precipitate quickly to its thermodynamic solubility. But it's possible to delay it is possible to delay onset of such a precipitation by adding addition of compatible so-called parachute, which is typically a polymeric excipient that effectively slows down the precipitation. And once again, when we're doing that, our motivation can be twofold. We either want to be as good as the originator, but maybe using a different excipient. That can be for different reasons. Either we discover throughout the development that the combination used by the originator is not the most stable one, or it has some unfavorable properties, or maybe the original composition is patented. Well, or on the other hand, we might be looking for a better solution. And again, that is applicable in whenever we are looking for separate bioavailable formulations. So here is just a depiction of the experiment on the Inform, how it looks like. So you can imagine what's happening in there. Typically, we're measuring concentration with a UV dip probe. And we like to run these experiments in bioequivalent media to mimic the environment in the gastrointestinal tract. And we start the experiment by addition of API pre-dissolved in an organic solvent, typically the MSO, but it can be something else in case it makes sense for the particular drug of interest. And this is a typical result that we get when we're looking for well, the best concentration where we actually want to run the experiments. So here, you can see that with increasing concentration, the precipitation is not only quicker, but it happens sooner. So again, this is a typical result that we might get from initial round of precipitation experiments on Inform. So once we have a nice plot like that, we know that what concentrations might be the best to run some screening of available excipients, and that's what we would typically do. So we already know how the precipitation curve, or how the concentration time profile looks like when there is no polymer added to the experiment, where it's just the API precipitating in bioequivalent media. And then when we add some polymers, we see that we are able to modify the precipitation kinetics, basically. So here, for instance, we see that in presence of these polymers, we get a prolonged so-called induction time, which is typically the time before the onset of precipitation. And once we are able to run these experiments, evaluate this induction time, we are once again able to plot result that might look like that. This is actually from a paper that we published already, where we did a screening of precipitation inhibitors on Inform. And here you can see that we were able to screen number of excipients. We evaluated induction time as relative induction times. That means relative value to an experiment where there was no polymer added to the solution. On this next slide, I'd actually like to show a nice case study that we had in our development, where we were trying to match the performance of the original drug product or of the formulation prepared by the original company. In this case, we were limited by IP, so we knew that we couldn't use the same excipient as the originator. So we wanted to basically screen a pool of options that we had available, and for that we used Inform. So first, we wanted to see how the API precipitates on its own in biorelevant media again. So you can see that is already quite interesting, even for the API itself, that it precipitates quite quickly, and it first seems to precipitate to some metastable form, and only then to precipitate to its stable crystalline form. So then when we added to the experiment the polymer that was actually used by the original, and I labeled here the original polymer, we saw that it really acts like a powerful parachute or powerful precipitation inhibitor, because now you don't see any precipitation for a course of 3 hours. So you knew that well, the originator chose well, basically. We had some ideas on what to use, based on some other properties of the excipient, stability, and so on. So we wanted to see how this excipient of choice performed in such kind of experiment, in precipitation experiment. So this is what we tried. Here I label it just polymer X because I cannot disclose what it was. But basically what we saw, you can see it right away that this polymer wasn't as good as the one used by the originator. Simply speaking, you see that the precipitation happens instantly. Probably this polymer is able to stabilize this metastable form, but still its performance is definitely worse than the one of the originator, or the one of the original polymer. So we were thinking what to do next, and we thought that it might be feasible to just add another excipient to improve the behavior here. So that's exactly what we did. We added another polymer, and we saw that finally by this combination we were able to match the performance of the originator in terms of precipitation and inhibition. And well, lastly, speaking about precipitation experiments, that's actually something that we run quite a lot on Inform. And the reason why we use Inform for that is because it has such well-controlled conditions for these experiments. And I'd like to show just a few pictures that illustrate, how important it is to have controlled conditions. So I showed you this picture before. I used it as a depiction of the precipitation experiment, so to explain how it works. But when you look at the Inform, you see that you would see that the API is not added to the solution by a sample syringe, but there's actually an inbuilt syringe in Inform, so it looks more like this. So the API is injected directly into the solution. And what we noticed in some of our studies was that position of the needle, let's call it needle depth, actually matters a lot when we want to run these types of experiments. And also there are some other conditions that might influence the precipitation kinetics, such as the speed at which the samples are added to the media. So here I just plotted a really simple experiment where we actually adjusted the speed at which the sample is injected into the media and evaluated so-called induction time. And you could see that with different needle valve speed, that is different speed of addition of the sample to the media, we get different induction time. So that's something that one has to be aware of, and it's important to know about this, not to base some conclusions of experiment that might be misleading in this way. For instance, if you would be doing these experiments by hand or manually. As I mentioned before, needle depth or position of the needle might also play a role in the outcome that we get from the precipitation experiment. So for instance, here I just plotted two needle positions that we tried just for our sake, just to see what's going to happen. And I plotted here over time scattered light or absorbance at 500 nanometers, which can be used as a direct indicator of precipitation. And you see that when we use the needle position indicator here by the red color, we see a nice profile with a distinct induction time and a precipitation phase. But when we change the needle position slightly, we see that a precipitation that is really hard to evaluate these data and hard to evaluate induction time from data like that. So again, that's something that is important to keep constant throughout the experiments when we want to compare or screen pool of excipients, for instance. And well, last thing that I wanted to show when it comes to repeatability is again something that you might think of, but I think it's nice to see it depicted again. Is how agitation speed or rotation speed of the stir might influence again precipitation kinetics. So here I ran the same experiment with the same setup using 3 different rotation speeds, and you can see that with higher agitation speed, I basically got a, again, earlier onset of precipitation, which I would evaluate as shorter induction time, and that might skew the results. That might skew the results that we get from screening of any excipients. So I wanted to show this slide just like as a heads up to really stress that it's important to have an instrument like that if we want to have precisely controlled conditions and not to introduce any flukes into our data.
Okay, so that was T3 and Inform, and now we are going to show or share some case studies using rainbow fiber optic probes together or alone and together with MacroFlux. And as you can see on this picture here, we actually used very old dissolution path that we inherited from our QC unit, and we equipped it with Pion fiber optics and also MacroFlux setup. And this enables us to run various experiments and different setups, and we will show them in next few slides.
So on this slide, we have a figure that represents a result that we obtained with using rainbow fiber optics. No MacroFlux was run in this case, only dissolution, and the detection was thanks to the rainbow fiber optics. And this is the product or the molecule which tends to precipitate, and we're trying to improve it, or we're trying to get rid of the precipitation and improve the product bioavailability or the bioavailability of the molecule. And thanks to rainbow instrument, we were able to capture here the profiles really nicely. And so you see here 4 different prototypes. Each of them has a different profile, and you see that at least 2 of these, they have some tendency to supersaturate slightly and then precipitate. And in the green one, that dissolves a bit slower, but then holds supersaturated. I would say the advantage here is that with the in situ probes, we don't need to filter the sample and analyze them later. Typically, if we want to do this by using regular dissolution bath with offline UV or HPLC measurement, we have to filter the sample, and then since it has a tendency to precipitate, we also need to dilute it immediately. So it's quite labor intensive to do it this way, and there is a possibility of human error, which you can skip the while or you can dilute it more or less. So in this way, we are skipping this step, which is relatively complicated, and still by using fiber optics we obtain quite useful result. And on this next example, we have a figure or dissolution graph where we're showing or comparing the result obtained by the pedal apparatus and rainbow fiber optics, which is the gray line here or points, and also black line, which are the data obtained by again USP2, but by standard filtration and HPLC or UPLC measurement. You see there is some slight difference between the two, but for initial investigation, this was good enough for us. You know, getting the data from fiber optics, because it was clear to us that we need to, if we run standard USP2 measurements with filtration and HPLC measurement, we needed to have the method developed first. But for some first prototyping, we used our fiber optics, and we were able to eliminate some some prototypes which clearly didn't work in the way we wanted, and then only then in the later stages of the project we developed the UPLC method and used the solution in more standard way. But for initial screening and scouting, this was perfect and rather easy to use. And now I will hand over the microphone to Teresa, who will describe or show you more case studies on use of MacroFlux.
The last instrument that I would like to introduce here is Pion's MacroFlux system that we use for dissolution permeation studies. So this instrument consists of a standard USP2 or USP1 vessel equipped with a UV dip probe. This part serves as a donor compartment, but what is really important here is this little add-on, which serves as an acceptor compartment. It is also equipped with a UV dip probe and also an overhead stir. And these compartments are separated by a membrane that is covered with lipids that basically serve as a biomimetic artificial membrane that can be used to simulate passive diffusion through biological membranes of any drug of interest or of any drug that permeates through passive diffusion. So when we use this instrument, we don't only get a dissolution curve, such as here, or dissolution data, but we also get some data from the acceptor compartment, and this is how a typical result might look like. From these data, we are able to calculate something called flux, which is basically a slope of a time concentration curve that is generated in the acceptor compartment. And you can see here on this expression that flux directly correlates to permeability and also to the concentration of the drug of the donor compartment. This is important to understand for the future result that I'd like to show. And you can also see from this expression here that this flux can be calculated from the slope in the slope of the time concentration curve in the acceptor compartment. Now, you might typically find in the literature that people are attempting to use this instrument for well, bioavailability predictions or relative bioavailability predictions. So I'd like to show just a few results from a published paper where this instrument was employed to study the effect of basically pH of dissolution media and its impact on flux or permeation rate of a compound of interest. So in this paper, the authors got these two results that corresponded to the conversion from pH 1.6 to passive and conversion from pH 4 to passive. Here you can see some dissolution data and also absorption data. From these, you can, of course, again calculate the flux. The authors claim that the simultaneous dissolution absorption measurements allow for more accurate in vivo predictions, and they were actually able to successfully predict what's going to happen in vivo just based on these flux measurements. In our development, we haven't really had a case where we would use this instrument to successfully predict or precisely predict the outcome of a bee study, but we did have some really interesting studies where we actually used the instrument to understand drug excipient interactions. So I'd like to show one of them here. To state a problem that we were solving, we had an original formulation that contains a poorly soluble drug. And we wanted to improve this formulation by reformulating into something else, either amorphous dispersion, lipid-based formulation, or whatever, to achieve a supra-bioavailable formulation. Here you can see a result from really simple dissolution experiment in phosphate buffer. It was actually generated on the MacroFlux device. And here we tested the formulations that we had available at the time. So we had the original formulation that dissolves really poorly. It contains crystalline drug micronized. Then we had a nanomilled crystalline drug. We also had a lipid-based formulation and an amorphous solid dispersion. So based on these results, or just these dissolution results, one would expect that if we were to test, let's say, the amorphous solid dispersion in vivo, we should theoretically get much higher availability when compared to the crystalline drug, right? So that was our theory. But let's have a deeper look at the data we were able to generate on MacroFlux. So let me start with these two formulations. They both contain crystalline drug. The difference is just in the size of particles. And when we looked at the flux, it made perfect sense. So for the crystalline drug, flux was low. For the nanomilled drug, the flux was higher, which corresponds to better dissolution in the donor compartment. So up to now, the data really did make sense. But when we generated the flux for the other formulations, for the lipid-based formulation and for the amorphous dissolution, amorphous solid dispersion, we noticed something really strange, and that is that the fluxes that we generated were surprisingly low, given the amount of the API that should have been theoretically dissolved in the donor compartment. And when we looked at the formulations more closely, we noticed that they actually contain one common excipient, which was this polymer, HPMC-AS. And after running further experiments, we actually identified that this polymer interacts really strongly with our API, and that might probably will interfere with the permeation of the API. And actually, really, in the real world, when we ran the study in humans, the clinical trial, where we wanted to compare the amorphous solid dispersion against the crystalline drug, we didn't see such a nice result. We still reached higher bioavailability, but we didn't really meet the expected target. And the best hypothesis that we have so far is was formed based on these experiments using MacroFlux, where we were able to show that the API polymer interaction, the specific interaction, may actually play against absorption of the API. So following up on this case study, we now can actually say that this instrument is really useful to study either compatibility or incompatibility with certain excipients or polymers, and we find it a useful tool to identify some interactions between API and polymers. So in our typical workflow, what we now use it for is to basically rule out any unwanted interaction between API and either polymer or other excipients. And here I just wanted to show one result where we are confident that there is no interaction between API and polymer. So these are some dissolution curves that were generated in presence of different excipients. And here are fluxes that were measured in the same experiment. And you can see that there is actually a nice correlation. So whenever we see improved dissolution, we also see slightly improved flux. So that means that the apparently dissolved API in the donor compartment directly translates into the higher flux measured in the absorption compartment. So here, when you get an experiment like that, we're confident that we can say that there is no interaction that might interfere with absorption of the drug. So to conclude, you could see that well, in our quest to understand what is happening with a dosage form inside a human gastrointestinal tract, we use a lot of pion instrumentation. We hope that we gave you a bit of an insight on how to use this instrument or how we use them in a generic drug product development. And well, if there are any questions regarding either T3, Inform, use of Rainbow, or the MacroFlux device, we are available to answer them, and we'll be happy to take them. And once again, we really appreciate the opportunity to speak here, and we hope that you enjoyed this webinar.
So thank you, Joseph. Thank you, Teresa. We have a few questions. So you showed a lot of precipitation type experiments and studies using different excipients. So do you see any interferences in the UV vis spectra caused by excipients or solid particles when using the fiber optics?
Yeah, so actually it's pretty good. The interferences are usually not a big issue. The only time to be concerned is when we work with excipients or polymers, more specifically, which can absorb light. So HPMCP, hydroxy propylmethylcellulose phthalate, or Soluplus, those are kind of problematic, and they tend to increase the response from probes. But otherwise, I think the interferences are not a big issue in most of the cases.
Okay, thank you. So I have another question here. So again, you showed that you're doing quite a lot of work on kind of solid dispersion technology. But what proportion of drugs, from as a generic manufacturer, what proportion of drugs are you working on that require just standard disintegrating dosage forms versus enabling technology?
That's a good question. I'm not sure we know the answer in presence, but we have it in our portfolio. We have it mixed. So we look at the standard approaches, and then enabling. I would say my estimate is 20% for enabling formulation, 20-30% for enabling formulations, and the rest for the standard approaches.
Okay, thank you. Interesting. You showed some slides on measuring intrinsic dissolution rates. How do those results compare to the more traditional large-scale apparatus?
Yeah, so I can answer it. We tried to compare some of our data across different apparatuses. Of course, the numbers are always a bit off. So a number that we get on intrinsic dissolution rate on Inform might be a bit different from other instruments. However, if we would be looking at the rank order of different solid forms, or at the ratios between them of measured intrinsic dissolution rate between them, then I would say Inform is quite comparable to other devices or more standard approaches.
Okay, thank you. Okay, I think I have one more question here. You mentioned early on in the presentation about pharmacokinetic simulations. Does that mean, do you use any kind of modeling software tools or PBPK or anything like that as a generic formulator?
Yeah, we do. Oh, I hope I can name the software here. But we're using TPROMs from Siemens PNC company to do some modeling. Again, it's not something that we would do for all of our projects that we have in development, but for those that require, this type of approach or that are maybe more complex, we try to employ it.
Okay, thank you. Well, I don't see any more questions coming in at the moment, so I think actually this will make a good time to finish up, and I'd like to thank you, Teresa and Joseph, for giving the presentation today, and I hope the audience found it interesting. But thank you, everyone, and we're going to close the meeting now. Thank you.
Thank you,
Goodbye.
everyone.
Thank you. Bye-bye.
Bye.